out of your day to come and share all of your knowledge with us, and even more so, we're appreciating you answering the question. So for the audience, if you go ahead and put your questions in the chat pod in the lower right, we will hopefully get to all the questions we have time for. So with that, we'll get started with Dr. Zhen for our first question. So, Dr. Zhen, so the whole blood stability experiment, does it require fresh whole blood or not? Sometimes it's difficult to get fresh whole blood unless the lab and clinic are located in the same building. Is it okay to collect the whole blood earlier and keep it refrigerated keeping it in a refrigerated condition?
Yeah, thanks for the question. So before I answer this question, I want to make a correction on my slide number 6. The percentage in the pie chart has typos. It should be 26% without precautions and 74% with precautions. So now let's get back to the question on whole blood stability experiment. So usually we recommend to conducting whole blood stability as soon as possible. But if you consider time is an issue, you may provide justification for not being able to conduct whole blood stability using fresh whole blood, or you can contact review division to control the correspondence. Thank you.
Great, thank you, Dr. Zhang. And we have another question for you. Could you please elaborate on what do you mean by autosampler stability? What does that imply?
Autosampler stability implies the processed sample at the injector or autosampler temperatures. Thank you.
All right, we have a third question for you, Dr. Zhang. If there is a back conversion, how do you stabilize the analyte?
The selection of stabilizer or enzyme inhibitor should be based on the mechanism of back conversion, and it should be validated in the method validation. There's no universal stabilizer or enzyme inhibitor for back conversion, so it should be case by case. Thank you.
Great, thank you so much. And we will send a couple questions over to Dr. Mao's. So for Dr. Mao, what is an SAS dataset?
So SAS dataset is a SAS transfer dataset that applicant usually provide in their end of submission, and it really contains information regarding the study sample concentration, the actual time point of the plasma sample collection for each subject, and their group number, their period, the sequence, and also the PK parameters that they narrowed.
Okay, great, thank you. Another one for you. Can you clarify the difference between reanalysis and reinjection?
So reanalysis usually means that this study sample is reprocessed from the beginning, whereas the reinjection is basically using the sample that are already in the autosampler, and then you reinject it again without any reprocessing.
Okay, great. Another one coming your way. So from your perspective, should the poor chromatograms have been included in sample analysis report to support sample reanalysis? Should they do that?
Yes. So as I mentioned in my presentation, even if you submitted if you have poor chromatograms, both the rejected and the accepted chromatograms should be provided, and it should be provided in modified, and it's up to you whether you would like to combine it together with your bioanalytical report, or you can also submit it as a separate document if you feel like this will if the a file size is too large, you can also submit it in a separate document. So it's up to you, as long as you submit it in module 5 in your application.
Great, thank you. And we have one more for you, and we'll switch back to Dr. Zhang. So Dr. Ma, for the ULOQ repeat, can you clarify on what markedly lower than ULOQ means? Is there any criteria?
So there's no set criteria for this, but think of this way. So if your original sample concentration is above, say, ULOQ, but then when you repeat it, your repeated concentration is only 50% of the LOQ even after correcting for the dilution factor. And then this will raise concern to us regarding the analysis of this specific sample. So under this condition, we'll ask you to explain why there's such a difference in the study sample concentration.
All right, thank you. Thank you so much. All right, for Dr. Zhang, we have another question. So is it mandatory to perform long-term stability analysis of one analyte in the presence of another analyte in the case of multi-analyte BE studies?
Its method validation guidance recommends for drugs administered as fixed combinations, the stability of the analyte should be assessed in the presence of the other drug. But you can also provide justification if you did not include another analyte in the long-term stability analysis. So you need to do more research to support that you did not include another analyte. Thank you.
Okay, great, thank you. All right, another question for you. So sort of a long question. Which metabolites should be taken into consideration from the perspective of back conversion through sample extraction from matrix and on instrument, for example, ion source back conversion, while developing a method or validating a method for bioanalysis, small molecules? So we have a follow-up with that. Should it be based on a type of metabolite or based on concentration of the circulating metabolite?
Metabolites with known or suspected back conversion issues should be always investigated. The evaluation should be based on both the concentration of circulating metabolites and the type of metabolite, such as acylglucuronide conjugated metabolite. In the method validation, the medium should include both parent analyte and its metabolites with potential back conversion issues at various concentrations. The concentration of metabolites used in the evaluation should be relevant to the concentration of circulating metabolites in the system. Thank you.
Thank you, Dr. Zhang. We have another question for you. Can you elaborate on ISR implications in addressing labile metabolite back conversions?
ISR is a useful tool to identify stability issues. Back conversion can lead to failed ISR or sometimes ISR meeting acceptance criteria, but with large difference between original and repeated values for many samples. If back conversion occurs during the storage, I want to emphasize during the storage, reanalyze the values of the parent drug in ISR samples are usually higher than the original values. But there are other mechanisms which can lead to ISR failure. So if you observe the unusual ISR results, so further investigation is needed. So another thing to consider is that failed ISR may not be caused by only one single issue. It can be caused by the combination of multiple issues, such as back conversion, degradation, or solvent used in the analysis process. So it is important to conduct a thorough investigation on field ISR. I also want to emphasize that when the matrix used in the DE study is plasma or serum, ISR usually is not able to identify stability issues if stability issues only occur in the whole blood. That is why whole blood stability is important when there is a potential stability issue for the analyte, especially in the whole blood. Thank you.
Thank you so much. And another one for you, Dr. Zhang. Is the effect of circadian rhythm usually considered during validation of bioanalysis methods for endogenous compounds?
So for bioanalysis part, we don't need to take consideration of circadian rhythm. But in the PP analysis, you may need to explain or justify circadian rhythm on your PP analysis. Thank you.
Great, thank you. And our next question is for Dr. Ma. If a sample was not reanalyzed due to an insufficient sample amount, should the original value be reported even though the reason for rejection was due to an internal standard variation?
So since the sample was identified for reanalysis due to, say, example, due to internal standard variation, it means the original concentration is not reliable without repeat. So in this case, the reported, if this is also defined in our SOP, the sample should be reported as missing value. And then, but in your bioanalytical report, you should specify that the original concentration you measured and the reason you identify it for sample repeat and the reason that reanalysis could not be conducted, which in your case is insufficient volume, and that would be okay.
Okay, great. Another one for you. For ULOC repeats, does the dilution factor have to be exactly the same as the one validated in the method validation?
It does not have to be exactly the same, but it needs to be within the range of the dilution factor validated in the method validation. So for example, if you validated twofold and also tenfold dilution in your method validation, and they meet the acceptance criteria, and then in your study sample reanalysis, you decided to use, say, fivefold dilution in your study sample, then this is, in general, would still be considered acceptable.
All right, great. Another question for you. When you mention reporting numerical raw data, do you mean peak area from all runs? Should this be included in the sample analysis report?
So the numerical raw data, it should include the data of the peak area or peak height for the drug and peak area or height for the internal standard, ratio of the peak area or height for the drug to the peak area or height for the internal standard, like dilution factor, if any, and corresponding concentration for each assay and reassay samples, calibration standard samples, and quality control samples. And this can be submitted either as a separate document or appendix of your bioanalytical report, or to be provided in the same document as the bioanalytical report. Is up to you. And this should be submitted in Module 5.
My apologies. I was on mute while I was asking this question. So we have one more question for Dr. Ma, and then we'll switch over to Dr. Zhang. So Dr. Ma, according to your presentation, unaccepted and accepted chromatograms should be reported. But the VMV bioanalytical method validation guidance requires you to submit a minimum of 20% chromatograms. If more than 20% chromatograms are included in the submission, are the unaccepted chromatograms still required to submit?
So yes, so even if the more than 20% of chromatograms are submitted, you are still recommended to submit both the accepted and also the rejected chromatograms for those samples repeated due to poor chromatograms. And this is to help the reviewer to verify whether the samples that you rejected due to poor chromatograms are rejected per the SOP.
Okay, great, thank you. Our next question is for Dr. Zhang. Do we need to perform parallelism for all endogenous samples during validation?
It's not necessary. Parallelism study is most useful when you use surrogate matrix. You can also provide justification if you think parallelism test is not necessary for your specific drug product. Thank you.
Okay, and another one for Dr. Zhang. Why do we need 3 replicates of the parallelism test?
We recommend 3 replicates of parallelism test in order to get information for inter-run precision and accuracy for parallelism test. So supposedly you should conduct run parallelism test samples in 3 separate runs. Thank you.
All right, great. Another one. If using a surrogate calibration curve, shouldn't there be some sort of a QC in surrogate used to ascertain the performance of the curve along with the HSQCs?
So I assume you are talking about study sample analysis. So for study sample analysis run, surrogate QC is not necessary because QC in authentic matrix cover the study sample, and the calibration curve standards can be used to ascertain its performance by comparing to their corresponding nominal values. Thank you.
Thank you. All right, another question for you, Dr. Zhang. What is the acceptance criteria for parallelism?
At this time, we don't have objective acceptance criteria to evaluate parallelism experiment, but I would say 5% is a good start. Thank you.
All right, another one for Dr. Zhang, and then we'll move to Dr. Ma. What software can be used for the regression subtracted standard curve? Can you use Excel?
You can use any software which has the function to do the regression subtracted standard curve. Thank you.
Okay, and for Dr. Ma, can you elaborate again on the need for multiple SOPs for study sample reanalysis as opposed to having one SOP on sample management?
So the SOPs the criteria for repeat analysis can be in different SOPs or in one SOP. For example, as I mentioned in my presentation, some applicants may have the acceptance criteria for chromatograms in one SOP, and then the wrong acceptance criteria in another SOP, and then the rest of the acceptance criteria for the rest of the repeat code in a separate SOP.
All right, thank you. A question for you. If a sample is qualified for repeat under poor chromatography reasons, and in the repeat, the same sample qualifies for repeat under IS variation, under which region should it be reported in the BE summary table?
So I see applicants do different ways. I think both ways are acceptable. So let me give you the two examples. So one example is that you can have one category saying that the sample is first repeated under poor chromatography, and then was repeated again under the internal standard variation. Or alternatively, you can also put this under the category for poor chromatogram, and then put say an asterisk on beside your the number that you reported, and put a footnote under the table saying that well, say one sample of these 5 samples repeated due to poor chromatogram was repeated again due to the internal standard variation. So as long as you make it clear in your submission, this will be considered okay.
Okay, great. And we have another question for Dr. Zhang. If the validated calibration range is needs to be changed, then is it required to prove stability again?
If the changed calibration range can be covered by the validated calibration range, then you don't need to prove stability again. But if it cannot be, if the changed calibration range cannot be covered by the validated calibration range, then you need to do a further stability test. Thank you.
All right, we have another question for Dr. Ma. Is it necessary to set limits for internal standard variation when stable isotope labeled analogs are used as internal standards?
So we recommend you to set limits even if you are using the stable isotope labeled analog for your internal standard variation. And you can refer to the guidance for industry evaluation of internal standard responses during chromatographic bioanalysis questions and answers for more details.
Great. Thank you so much for this long and very beneficial question and answer session. We appreciate your insights into your presentations. It's great to hear from the FDA. And with that, we're